This presentation will cover the action potentials of cardiac contractile cells and will be presented by myself, Haley Rhodes, a graduate student at Grand Canyon University. In this presentation, we will cover the vocabulary of action potentials, contractile versus pacemaker cells, the action potential of contractile cardiac cells, and lastly, we will take a look at a study scientists completed with a connection of magnesium, TRPM7, TRPM6, and their effects on the action potential of cardiac cells. When we take a look at action potentials, it is important that we have an understanding of the vocabulary that is involved. Action potentials are quick changes within the membrane that happen at a very short period of time. The graph on the right depicts these changes. Potentials will happen when there is any change in the resting potential. When the cell membrane is at a resting potential, there is no electrical signals being produced. This particular cell's resting potential is at about -70 millivolts. Not every cell has the same resting potential. Depolarization will happen when there is a change in the resting potential. This change is seen here in the graph as the line begins to approach 0 millivolts. Depolarization typically begins slowly until the threshold potential is reached. Threshold potential is here in green. And then it happens very rapidly. For this graph, the threshold potential is about -50 millivolts. After depolarization has a rapid increase, it'll reach the peak potential. In this graph, we can see that the peak potential is about a +30 millivolts. Repolarization occurs after the peak potential threshold is reached, which we can see here in the downtrend, with the goal of going back to the resting potential. In addition, the potential can overshoot the resting potential, and this overshoot is known as hyperpolarization. Cardiac muscle cells are comprised of two main types of cells, autorhythmic and contractile cells. Autorhythmic or pacemaker cells are few in number, but can provide the action potentials for contractile cells. Contractile cells will receive the action potentials and contract the cardiac muscle. Pacemaker cells use the sodium ions to initiate the action potential, but once the potential reaches the threshold, calcium ions are used to bring the potential to the peak, while contractile muscles cells use sodium ions to reach its peak potential. Contractile muscles use calcium ions. During depolarization, which we will talk about on the next slide. During repolarization, pacemaker cells' potassium ion channels will slowly begin to close, causing repolarization to happen at a slower rate. Pacemaker cells will have a short hyperpolarization, causing a special sodium ion channel to open to send the membrane toward the threshold potential again. These channels are known as funny channels. And while this causes pacemaker cells to have no true resting potential, leaky potassium ion channels in contractile cells cause the resting potential to be at -90 millivolts. Cardiac contractile muscle cells have a unique action potential. On the right, we can see what this action potential looks like. There are 5 main phases through this potential. The resting potential is represented as stage 4, which can be seen here on the left-hand side of the graph, as well as on the right-hand side. The resting potential of contractile cells is typically around -90 millivolts. This very negative resting potential happens as a result of potassium ion channels being open and potassium leaving the cell. When the stimulus is received from pacemaker cells, a rapid depolarization takes place. This is represented by phase 0, which we can see here. Sodium ion channels open and potassium channels close, and the sodium ions rapidly move into the cell. This rapid depolarization is often seen as a fast response. Once the peak of the potential is reached, sodium channels close and potassium channels begin to open for a short time, causing the start of repolarization, seen here at phase 1. At phase 2, the membrane will start to open L-type or long-lasting calcium ion channels. Calcium ions will move into the cell, tremendously slowing the repolarization process. This is also known as a plateau phase. At phase 3, which is here, calcium channels will become inactivated and potassium channels will continue leaving the cell, allowing the action potential to trend towards the resting potential. This significance of this repolarization process distinguishes contractile cells from the action potential seen in muscle or nerve cells. The effective refractory period, or ERP, occurs between action potentials. During ERP, stimulation from a neighboring cell will not produce a new action potential. The purpose of the ERP is to allow the heart to fill with adequate amounts of blood before contracting. At high heart rates, this cannot happen. In 2021, Guignana and associates looked at how action potentials can change in the presence of magnesium ions. These scientists previously studied magnesium ion effects on cardiomyocytes. This current study is looking at how TRP receptors play a role in magnesium ion influxes. TRP receptors are found throughout the body, and there are many types of TRP receptors. In cardiomyocytes, the known TRP receptors to play a role in electrophysiology are TRPC1, TRPC3-6-7, TRPV4, TRPM4, TRPM7, and TRPP1-2. In previous studies completed by these scientists, they noticed how TRPM6 and TRPM7 channels are sensitive to magnesium. These channels are activated when they are in low concentrations of magnesium within the cell. Recent studies have shown that TRPM6 and TRPM7 have been co-expressed. In addition, channels that allow influxes of magnesium ions are also known to be permeable to calcium ions. The goal of this study was to see how currents of magnesium ions, TRPM6 and TRPM7, affected pig cardiomyocytes. The first step in this study was to determine the presence of TRPM6 and TRPM7 in the pig cardiomyocytes from all 4 chambers of the heart. The cells were stained and underwent immunofluorescence. When looking at the graphs on the screen, we can see the totals of TRPM7 on the left in green here, and TRPM6 in red on the right here. The x-axis is labeled on both graphs with LA for left atrium, RA for right atrium, LV for left ventricle, and RV for right ventricle. We can see that when comparing the quantities of these 2 channels, that there are more TRPM7 channels present. In both TRPM7 and TRPM6, we also notice a trend in increasing amounts from the left atrium to the right ventricle. Next, we will take a look at how magnesium currents affect the action potentials. The scientists used a whole cell voltage clamp to record the total currents. The cells were then activated or inhibited with solutions. Cells were internally dialyzed with solutions containing magnesium ion concentrations of 0.8 millimolars or were given magnesium ion free solution. The cells were then extracellularly perfused with either physiological levels of magnesium, which is at 0.9 millimolars, or high levels at 7.7 millimolars. In previous studies, the high levels were known to suppress magnesium ion currents. When taking a look at the results, you should first take note that the graphs on the left represent the 0.8 millimolar internally dialyzed magnesium, which is here on this left. And then on the right is the free magnesium ion solution with 0 millimolars of magnesium ions. Graphs A and B, and the black lines throughout these graphs represent the control, or the 0.09 millimolars of magnesium ion in the extracellular solution, and the red will represent the high level magnesium extracellular solution. Graphs A and B at the top here represent the whole current obtained by using voltage ramps. Graphs C through F are summaries of the action potentials. The cells that received the internal dialyzed solution of 0.8 millimolar of magnesium did not see much change in the potentials between the control and the test solutions, while the magnesium-free internal solution had a prolonged repolarization rate in the high-level solution, which we can see in graph D. When looking at this information together, it is modulated by outward currents, which are activated in free magnesium solution but suppressed in the high solution. Since the current was absent in the 0.8 millimolar solution, it is possibly due to the TRPM7 and TRPM6 channels. In conclusion, the results suggest that there is a presence of TRPM6 and TRPM7 proteins in cardiomyocytes. The immunofluorescence showed that they were present, and in addition, the difference in currents between the dialyzed cells showed no current changes in the 0.8 millimolar solution, while there was a prolonged repolarization in the free magnesium and high-level solution. These conclusions further solidify findings that magnesium ion sensitive channels are due to TRPM6 and TRPM7, and that they may have a role in the cardiac magnesium sensitive channels modulating electrical currents. These are the references I used. The first one is the journal article with the study. And then I use Dr. Kaplun's article on non-pacemaker action potentials, and then the book that we're using in this class on human physiology. Thank you for watching this presentation.